Assalamualaikum and hello everyone. In this video, we are going to discuss about strategies to improve stress tolerance in rice plants. This is our topic. Rice plant Oryza sativa produce an important staple food. It is crucial that this plant can thrive against stress such as drought and salinity. Explain several strategies to improve such stress tolerance in rice plant by giving example. In our video, we have 5 content, which is number 1, introduction, number 2, objectives, number 3, strategies to improve stress tolerance in rice plants, number 4, effects of improvement stress tolerance in rice plants, and number 5, conclusion.
Hi, I'm going to talk some interesting facts about rice plant. First of all, the scientific name of rice plant is Oryza sativa. Rice belongs to the genus Oryza and family Poaceae. There are primarily 2 species of rice cultivated for human consumption, which are Oryza sativa and Oryza glabrima. As we know, rice is a monocotyledon plant and the main source of food, which is carbohydrates. Rice is grown in a wide range of ecosystems, including flood and drought-prone environments, which is Asian countries such as Vietnam, Thailand, Bangladesh, Philippines, and Japan. Globally, more than 1/3 of the world's population consume rice as a main staple meal, and a large majority of people, particularly in Asian countries, rely on it for 18% of their daily calories needed. However, rice is very sensitive to salinity stress and is currently listed as the most salt-sensitive cereal crop. Salinity is a general term used to describe the presence of elevated levels of different salts such as sodium chloride, magnesium and calcium sulfates, and bicarbonates in the soil and water. Here are the objectives of this topic. First, to identify the strategies to improve stress tolerance in the rice plants. Second, to identify the effects of the strategies towards rice plant.
So now I will be talking about the conventional breeding. The classical strategy to obtain crops more tolerant to drought and salinity is through breeding. Conventional breeding is a method to develop new plant varieties by the process of selection and seeks to achieve expression of genetic material which is already present within a species. It is more powerful and efficient method of enhancing salt tolerance. However, the success rate of conventional breeding approaches is less. Plants tolerant mechanism is a complex phenomenon and depends upon psychological and genetic responses. There are a few factors for determining salinity tolerance, such as threshold and slope. So threshold indicates the highest permissible salt level without reduction in yield, while slope indicates % reduction in yield per unit rice in salt level above the threshold. So conventional breeding relies primarily on selection using natural process of sexual and asexual reproduction. Conventional breeding happens when 2 parents are crossed and segregating generations are screened for the trait of interest. Conventional breeding is achieved by crossing together plants with relevant characteristics and selecting the offspring with desired combination of characteristics as a result of particular combination of genes inherited from the 2 parents. So, conventional breeding has been used for production of salt-tolerant rice varieties through 2 basic methods. The first is to generate a breeding population that is highly variable for salt tolerance, and the second involves selection among the segregating progeny for individuals that combine most of the very useful traits and high degree of salt tolerance. So as you can see from the picture on the right, the donor variety have high grain quality while the recipient variety have high yield and high disease resistance. The donor variety have the desired Q gene, hence the donor variety cross with the recipient variety, producing a plant with a higher grain quality, high yield, and higher disease resistance. And just like the picture on the left, where it shows that this plant on the left has virus resistance, and while plant on the right has high yield resistance. And both of them cross and producing a high virus resistant and high yield crop. Furthermore, is the general steps in conventional breeding. There are 5 steps. Firstly are objectives. Breeding objectives depend on the species and the intended use of the crop to be developed. Example, the producer grower will cross crop profitably for the purpose to produce crop that is drought and salinity resistant. Secondly are creation and assembly of variation. Assembly of the required genetic variation for initiating the breeding. Example, artificial crossing of appropriate parents. Thirdly are selection and evaluation. Selection of the available or created variability to identify individuals with desired combination of genes or genotype. Evaluation which crops will be commercially grown. Evaluation which one of the crops will be commercially grown. For the 4th steps are certification and cultivar release. In countries with more advanced agricultural operations, there exist standardized and approved protocols for releasing new cultivars and growers. And then for the 5th steps are multiplication and distribution. Certified seed is multiplied by certified seed growers contracted by independent breeders and seed company to mass produce release cultivars for sale to increase. Moreover, I will be talking about the example of plant that involve in conventional breeding. Locally plant. Locally plant has been recognized for providing more salt tolerance when compared to other cultivar, and thus it is used as a highly potential salt tolerant donor. In this cultivar, a major QTL saltong was identified, which is located on chromosome 1 and involved in salt tolerance at the vegetative stage. This QTL causes 64 till 80% of the phenotypic variation under saline condition, which has also been confirmed in other varieties of rice such as Nona Bokra. Another rice variety is FL478, or its name is IR66946-3R-178-1-1. It is derived from crossing between Pokali and IR29. It has been promoted as an improved donor for breeding programs, as it has a high level of sealing stage, salinity tolerance, and is photoperiod insensitive, shorter and flowers earlier than the original Pokali. FL478 also maintains a lower sodium ion and potassium ion ratio than both the parent lines. Based on its ability to tailor well and maintain high potassium content under salinity stress, FL478 appears to be a good candidate for salinity stress tolerance in rice, particularly at the vegetative stage of growth. Last but not least is the advantages of conventional breeding. So, advantage of conventional breeding are increasing the availability of genetic resources for crop improvement, low cost compared to the other types of breeding technique, and lastly, are able to develop a disease-resistant crop.
Now we are going to talk about another strategies to improve such stress tolerance in rice plant, which is marker-assisted selection. We will discuss about definition, types of markers, how do we select the genes based on the criteria that we want, basic marker-assisted selection procedure, and the advantage of marker-assisted. What is marker-assisted selection? MAS. MAS is the process of using morphological, biochemical, or DNA markers as indirect selection criteria for selecting agriculturally important traits in crop breeding. It is a tool that can help plant breeders select more efficiently for desirable crop traits. For example, salt tolerance is a complex trait, so the quantitative trait loci, QTL, related with the salt tolerance have a significant role in understanding the stress response and generating stress-tolerant plants. So, to identify this trait, we can use the marker-assisted selection as a method. Next is the type of marker in marker-assisted selection. There are 4 types of marker, which is morphological marker. These markers are often detectable by eye and by simple simple visual inspection, leaf sheet coloration, height, grain color, aroma of rice, and etc. Second is biochemical markers. Biochemical markers is a protein that can be extracted and observed. For example, isozymes and storage proteins. Third is cytological markers. It is the chromosomal bending produced by different strains. Lastly, the type of marker in mass is DNA-based or molecular markers. It is a unique gene, DNA sequence, occurring in proximity to the gene or locus of interest. It can be identified by a range of molecular techniques. How do we select the gene based on the criteria that we want? Basically, there are many methods that we can use to identify the genes or QTLs. First is traditional based cloning. It is the process of identifying the genetic basis of a mutant phenotype by looking for linkage to markers whose physical location in the genome is known. Second is combination of genetic mapping. It is the process of determining the location of genes in chromosomes. And third is expression profiling. It is measures mRNA levels and showing the pattern of genes expressed by a cell at the transcription level. Other than that, we have the basic procedure in marker-assisted selection. The basic procedure for conducting mass with DNA markers is first extract DNA from tissue of each individual or family in a population. Second, screen DNA samples via polymerase chain reaction (PCR) for the molecular marker linked to the trait of interest. Third, separate and score PCR products using an appropriate separation and detection technique. Fourth, identify individuals exhibiting the desired marker allele. Lastly, combine the marker results with another selection criteria, for example, phenotypic data or other marker results. Then select the best fraction of the population and advance those individuals in the breeding program. The polymerase chain reaction (PCR) is used to make copies or a specific DNA sequence. Then we have the example of molecular marker such as random amplified polymorphic DNA (RAPD), simple sequence repeats (SSR), and restriction fragment length polymorphic (RFLP). Apart from that, we have the advantages of marker-assisted selection. The advantages of mass are simpler than phenotypic screening, especially in the case of complex traits. Other than that, the selection can be carried out at the seedling stage. Next is the simple plants can be selected. Both homozygotes and heterozygotes phenotype can be identified. Lastly, reduction in the space required for breeding as only selected germplasm is propagated.
Now we are moving to the next way to improve stress tolerance in rice plants, which is genetic engineering. We will discuss about the meaning, what related to stress tolerance, how the genetic engineering works, example, and also the benefits. What is genetic engineering? Genetic engineering and genome editing are plant breeding processes that allow plant breeds to introduce genetic modification techniques that utilize modern biotechnology. How is rice genetically modified? Rice genetically modified by transferring genes from the soil bacterium Bacillus thuringiensis into the rice genome. Genetically modified BT rice is resistant against Lepidopteran pest damage and thus less required the amount of insecticide used. So, what is the example of rice that genetically modified? The example is golden rice. Golden rice was developed in the 1990s to help improve human health. Rice is a good source of vitamin B, such as thiamine and niacin, but is poor in pre-vitamin A. So, golden rice was genetically modified to be a fortified food grown and consumed in developing countries where vitamin A intake is deficient. How genetic engineering can improve stress tolerance in plant? Several reports showed that manipulating genes that control the production of low molecular weight metabolites such as proline, which is an essential amino acid in plants, improves plant tolerance to abiotic stresses, including salinity and drought. There are more benefits in genetic engineering. Some of them are: first is enhanced nutritional value. Second is help in disease and drought-resistant plants that require fewer environmental resources. And the third one is less use of pesticides.
Next, effect of improving stress tolerance in rice plant. The effect of improving stress tolerance in rice plant are reduced negative effects of stress leading to be better growth and yields in crops growing in poor conditions. Conclusion: there are three strategies to improve stress tolerance in rice plant, which are conventional breeding, where two parents are crossed for the trait of interest. Second strategy is a marker-assisted selection. It have four types of markers in marker-assisted selection, which are morphological markers, biochemical markers, cytological markers, and DNA-based or molecular markers. And lastly, genetic engineering by manipulating genes that control the production of low molecular weight metabolites such as proline. So, by improving stress tolerance in rice plant, we can reduce negative effects of stress, leading to better growth and yield in crops growing in a poor condition. That's all from us. Thank you and bye.